Welcome friends in science lecture of standard 9th lesson 5, the fundamental unit of life. In this particular lecture we are going to start with plasma membrane or cell membrane. That's a new topic. Plasma membrane is an outer covering of the cell. It organizes the cell organelles inside. That means all the cell organelles, what we call as an cytoplasm containing the fluid inside, these all organelles like mitochondria, Golgi apparatus, lysosomes, in plants, plastids, and all these organelles including nucleus which is in the fluid is comprised by a membrane known as an plasma membrane. So, this particular plasma membrane is an outer covering of the cell from its external environment. In case of plants, we have one more external outer covering, that is cell wall. We are going to learn later on, but this plasma membrane in animal cells especially is an most outer covering which comprises the cell organelles and its fluid including the nucleus inside it, right. So, it is dealing with an environment directly, that means it is surrounding its surrounding. So, it also allows and permits the entry and exit of some material in and out of the cell. Obviously, the exchange of material which is done by the cell is obviously done by this membrane, whether it is in the case of gaseous molecules, whether in the case of liquid molecules, whether in the case of solid molecules. So, it allows, that means it allows or permits the entry and exit of the material. It also prevents movement of some other materials. That means it does not allow certain materials which are either harmful or not required, or it has already thrown outside such materials which are not allowed back inside, rather prevents this kind of material to enter inside the cell. The plasma membrane is therefore called as an selective permeable membrane. Why this word selective is there? Because it does not allow all kind of materials to enter inside, first thing. Secondly, it prevents the material which are either harmful or not required for the cell. So, it can be called as a selective permeable membrane, or maybe another word may be semi permeable membrane. Substance like carbon dioxide and oxygen can move across the cell membrane by the process of diffusion. So, as you are able to see in the screen, this word as in diffusion is from higher concentration to lower concentration. That suggests that carbon dioxide is basically what happens, that a glucose is being broken down inside the cell because of the liberation of the energy in the form of ATP, and when it is broken down, what is an waste substance which is produced is carbon dioxide. So, carbon dioxide concentration is supposed to be decreased by the cell, so it is throwing out the carbon dioxide outside the cell, and what it is taking inside is oxygen. So, this is an exchange of gas which is happening by the process of diffusion. Water also obeys the law of diffusion, and the movement of water molecules through such a selective permeable membrane is called as an osmosis. So, the word specially used for water from lower concentration, that is from dilute portion, that is having a more amount of water to the less amount of water, such kind of transfer of water is also done by this particular membrane which we understand as plasma membrane or cell membrane. So, this process of water diffusion is nothing but osmosis. The plasma membrane is flexible and is made up of organic molecules. You are able to see on the screen also, it is formed by a double layered, that is on the top layer and lower layer. The round balls that you are able to see is hydrophilic. Hydrophilic means these are the ends which are affected towards the water molecules, and the other ends which are not affected, which is called as an hydrophobic. So, this particular membrane is made up of a quite flexible material which can where you see our skin cells or our muscle cells also are quite flexible by its nature. They are all made up of plasma membrane. If it is made up of pure cell wall, then it is very hard. Outside the plasma membrane, if it is made up of cell, you are able to see in case of wood, they are very hard. So, it is flexible, made up of lipids and proteins. So, you are able to see a box like thing between this particular lipid molecules. This box like thing is nothing but protein. Proteins are responsible for this exchange of things for a particular thing, that if for to bringing the particular things, yeah, any kind of messages to inside the cell, these proteins are useful. Flexibility of the cell membrane enables to engulf food in case of amoeba. So amoeba is a single cellular organism. It has got only plasma membrane, does not have any cell wall because it is an animal cell, and it is like a water droplet with no boundary, and it's a kind of a boundary which is quite flexible. It moves due to its flexibility in the membrane, and continuously, like a drop is traveling, like a river is traveling, it is continuously traveling. So this amoeba and engulfs the food inside. That means whatever it comes in its way in form of food, it forms pseudopodia and takes it inside. It engulfs. Engulfs means to take inside. So it take inside the food by the help of an plasma membrane. It covers up the food inside. So these are many different kind of properties which the membrane is having, and we have learned certain things, that it is flexible, it is made up of lipids and proteins, it is capable of engulfing food inside, it is useful for exchanges of gases which we call as in form of diffusion, it is in the form of an exchange of diffusion in case of water, which is nothing but osmosis, and therefore it is called as an selective permeable membrane or semi permeable membrane. We will take a new topic after this, is osmosis. The word osmosis, which has occurred in this particular chapter, as we have already learned, the process by which the molecules of solvent tend to pass through a semi permeable membrane from a less concentrated, that is dilute solution, into more concentrated one, is called as an osmosis. I will explain you this definition as a process in which molecules of solvent, that means there are two things, solute and solvent. The solvent is a material which makes the solute soluble inside. So if I take a salt and put inside the water and stir it up, you say it's a salt solution, but the solvent is water and the solute is salt inside. Now if I keep the less amount of salt, we say it's a dilute. When if I take very large amount of salt and go on putting inside and make it very, so what we use word as in concentrated. When we take sharbat and all, also we take it either in the form of dilute, that means less amount of solute, or more amount of solute, that means it is more concentrated. Here the solution is a solvent is one which is capable of passing from a semi permeable membrane from less concentration, from a less concentration means dilute means more amount of solvent. Like if I say that salt is quite dilute, that means what we understand, that salt quantity is less and more amount of water. So more amount of water capable of passing through less amount of water, and this kind of passing of solvent molecules is known as an osmosis. Now there are three kind of osmosis. One is called as an endo-osmosis, another is called as an exo-osmosis, and third one is an equilibrium, that is water equilibrium. Osmosis word is specially used for water molecules only. Endo-osmosis, in the medium surrounding the cell, has an higher water concentration than the cell, meaning the outside solution is very dilute and the cell gains water by osmosis. Such a solution is known as an hypotonic solution, and the process is called as an endo-osmosis. If I will explain you that what happens, that for an example, if we take a resin means dry grapes, kishmish. So if you take this resin and put inside the water, now what happens, if you take a resin and directly eat it, you feel it very sweet by its nature. That means it has got very dry by its nature, just because it has got very less amount of water. It's a cell and it has got a very less amount of water. If I put it inside the water, what happens, the outside medium is completely more water and very less amount of salt. So here the water concentration is very high and the solution is very dilute, and inside the cell, that is resin, the concentration is very high and the water is very less. So what happens, the water starts passing from higher water concentration to lower water concentration, and slowly you see, if you put it at the night and see in the morning, you will see this resin has got swollen up with a water filled inside. So this process is called as an endoosmosis. Endoosmosis, the water is getting inside the cell. In the same way, exactly opposite to it, the medium has a lower concentration of water than the cell, meaning it is a very dilute concentrated, sorry, it is a very high concentrated solution. The cell will lose water by osmosis. Such a solution is known as an hypertonic solution, and the process is called as an exosmosis. Now again, in this particular define, what I said is a lower concentration of water. That means now the solution is very concentrated. You have put a very high amount of salt solution. Suppose if I take now pure grapes, a normal grapes that is available in the market, and put inside a very high concentrated solution of sugar or salt. Suppose if I take a very high concentrated solution of sugar, now what is going to happen, that the grapes, that is a fresh grapes that is available, which are already in the big green color grapes, which are already in the swollen condition, if I put inside a very strong sugar solution, here now you see the outside solution, that is solvent, is very highly concentrated. That means less amount of water, and the grapes itself contain a higher amount of water, that means less concentrated. So the water is going to come out from inside the grapes to outside the medium, and it is going to get empty with the water. That means it is going to get wrinkled or dry up, right? So this kind of process is, the solution is called as an hypertonic, and the process is called as an exoosmosis. Exo means outside, bahar nikalna, right? So it is going to get an exoosmosis by its nature. Now if I take in normal condition, if we drink water and the cell and our blood is having an equilibrium, so it does not either lose the water tentatively continuously, or it is continuously does not gains the water. So it is generally in the equilibrium condition, or tries to attain the equilibrium condition. And this type of when the inside of the cell and outside of the cell, both the areas, if you see, the water is almost in the same concentration, so neither the water is going to go inside the cell, neither the inside water is going to come outside the cell. Such kind of no kind of movement of water is called as an water equilibrium, and the solutions are called as an isotonic solution. Inside and outside, they are called as an isotonic solutions. Fine. We take some example of I'll show you one experiment of an egg. Now if you see, egg has got an shell that is made up of calcium carbonate, CaCO3. Like, so calcium carbonate egg shell is there. First of all, if you see, it's a covering. Now to break out this covering, we have to put inside the vinegar and allow it for 3-4 days to remain. Now after making it remain for 3 to 4 days, we will able to understand the eggshell has got decayed or reacted with the solution as vinegar and has got decayed completely. Now if you see properly, the inside the cell is still intact, just because it is been covered by plasma membrane. Fine. Now if we weight the cell and see on putting on the weighing machine, we will see it is approximately 97 grams, and this 97 grams or whatever the weight it comes, so this particular egg is having this much of weight. So what we are going to do furthermore, we are going to make one hypertonic solution and one hypotonic solution. That means more amount of water and less amount of water. Now one egg we are putting in the water, and the second egg we are putting in an hypertonic solution, maybe made up of salt, or maybe some kind of some concentrated acid, dilute acid, maybe hydrochloric acid. We can't take much concentrated acid, a dilute acid of hydrochloric, or maybe a salt solution. We will see in both the cases. In the first case, we will see that the water is getting inside and the egg is getting swollen up, right? And when the egg is getting swollen up, the eggs become heavy and gains heaviness and settles down inside the beaker. And in the second case, you are able to see the water is being lost in an hypertonic solution, and the water is lost, so egg shrinks and the size becomes very small. So the first case is endoosmosis and the solution is hypotonic, and in the second case it is an exosmosis and the water is hypertonic by its nature. So, from this particular experiment, the importance of plasma membrane is also understood, and second way, the exosmosis and endosmosis. If I put the egg in a normal solution where inside and outside pressures are same, it will be in isotonic form. Fine. Thank you for this particular lecture.